Okay, so here we are in section 3.3, 3.4, and 3.5. I've combined these sections because there's not a whole heck of a lot that we need to cover for these. The whole gist of this is we are going to be learning about the cell membrane and then how things actually move in and out of the cell membrane. Okay, so the cell membrane, if you recall, surrounds the cell. So we're going to look at how it's made and how that actually enables the cell to move things back and forth. So our key concepts: the cell membrane is a barrier that separates a cell from its external environment. The materials move across the membranes because of concentration differences, and cells use energy to transport materials that cannot diffuse across a membrane. Our vocabulary: we've got cell membrane, phospholipid, fluid mosaic model, selective permeability, passive transport, diffusion, osmosis, isotonic, hypertonic, hypotonic, facilitated diffusion, active transport, endocytosis, phagocytosis, and exocytosis. All right, so our main ideas. We have cell membranes are composed of two layers of phospholipids, and materials can pass through the cell via passive transport or active transport. Our essential questions. So what you absolutely need to know by the end of this PowerPoint is what are the structure and function of the cell membrane. Second part is what is passive transport and some examples of it, and then what is active transport and some examples of it. So first things first, our cell membrane. We're going to talk about its structure first. So what is it made of? Well, cell membranes are composed of two phospholipid layers. We call this two layers of phospholipids the phospholipid bilayer. Now you've heard of lipids before, fats, right? We learned about them when we learned about macromolecules. Phospho, because it's phosphorus, right? Bilayer, because we have two layers, so bilayer. Now, what is a phospholipid? If we look at a phospholipid and we actually look at the structure, we have two parts to it. We have this little round part on the top, we call that the polar head. And then we have these little tails down at the bottom, the straight lines. Those are the nonpolar tails. The heads, we say, are water-loving. They are hydrophilic, water-loving. The tails are water-fearing, hydrophobic, okay? And you can actually see over here on the side how this phospholipid bilayer is set up. We have the heads all facing in one direction here, and then down there on the other side, the two layers are facing in opposite directions, so that the tails are all in the middle together. Okay, in our cell membrane, we also do have other molecules such as proteins, carbohydrates, and cholesterol, and these act as little channels or little areas where other things can move through. So we're going to talk about that too. We use this model called the fluid mosaic model to describe the membrane. All that means, if you've heard like of a mosaic, like mosaic tiles, okay, are all these little pieces of different colors put together to make a picture? Well, we call it fluid because the individual phospholipids and proteins move side to side in the layer, kind of like a liquid, okay, but it's not a liquid. They kind of move, you know, back and forth and back and forth. And mosaic because those patterns, like tiles, okay, is a pattern produced when you look at it. You've got these little phospholipids with carbohydrates and proteins scattered throughout, so it kind of makes like this little random pattern. Some of the functions of the cell membrane. Well, it has two major functions. It forms a boundary between the inside and the outside of the cell. You know that. It surrounds the surface of the cell. So it forms a boundary from the inside to the outside. Then the other main one is what it controls what can move in or out of the cell. So it doesn't let everything in. We call the cell membrane selectively permeable, or sometimes they refer to it as semi-permeable, which means that some molecules can cross while others cannot. So it keeps things from crossing. It allows some things to go through, keeps other things out. This is super important for you to remember. The cell membrane is a phospholipid bilayer, and it is selectively permeable, which means it allows some things to pass through and keeps other things out. Now, materials can move through the cell membrane in several different ways, and there's two categories of transport. We have passive transport and active transport. And then we're going to talk about some examples of these as well. But passive, if you think about the word passive, you're just kind of like sitting by and letting things happen, okay? Same thing with our cell. Our passive transport does not require the energy from a cell. It's just sitting back, letting things go back and forth. It's passive, no use of energy. Okay, in active transport, we actually require the use of energy from the cell, so it actually has to be actively moving something back and forth. So remember, passive, sitting back, relaxing, letting things go back and forth. Active, actively moving something using energy. So we have three types of passive transport. We have diffusion, osmosis, and facilitated diffusion. In diffusion, molecules move from an area of high concentration to an area of low concentration. And you can kind of think about this, like if you had a bottle, like let's say some kind of room freshening spray, okay? If you spray that, what's going to happen is eventually you're going to start to smell that smell throughout the entire room, because the molecules from the can are moving from an area of high concentration inside the can to an area of low concentration. They're going to spread themselves equally throughout the room, and that's diffusion. So in diffusion, molecules move from an area of high concentration to an area of low concentration. Then we have osmosis. Osmosis is basically the same thing. It's just the diffusion of water. And again, it's going to move from an area of high concentration to an area of low concentration, okay? So osmosis is the diffusion of water molecules across a semi-permeable membrane. And again, we just use a special word because it is water. Now, one of my former students could always remember this by saying it's H2osis. That worked for him. So however you can remember, osmosis is the diffusion of water. So how does osmosis actually work? Well, if we look at cells, we can put cells into three different kinds of solutions. We can put it in an isotonic solution, hypertonic solution, or hypotonic solution. Now, in all of these different solutions, the water is going to move one direction or the other. If we put it in isotonic, the water is going to move in and out of a cell equally. Iso for equal. So as quickly as it's going in, it's coming out at the same rate. Okay? Equal amounts of water enter and exit the cell. Our cell will always look like a normal sized cell. Now, if we put a cell in a hypertonic solution, what happens in a hypertonic solution is the water actually moves out of the cell, okay? And when it does that, it shrinks it all down, causing it to become kind of jaggedy looking. And if you look at like that, you can think like in hypertonic, you can kind of think like hyperactive, right? Hyperactive, this big ball of something moving around real fast. It kind of looks like, oh, some kind of energy going on. So hypertonic, water is moving out of the cell, shrinking it down. Now, if we put the cell into a hypotonic, think hypo makes it get really big, like a big O, okay? Because water is going to move into the cell. And in animal cells, so much water can actually move into the cell that it can actually cause that animal cell to burst or explode, killing that cell. So hypo, water moves into the cell, causing it to get really big like that. Oh, hypo big, okay? Hyper, crazy, little squiggly. All right, now some molecules need a little help to move through the cell membrane. Now it's still part of passive transport, but it needs a little bit of help. And so remember, I told you at the beginning, there's these little proteins in between those phospholipids on our cell membrane. Now those proteins have two sides to them, and they kind of make a tunnel, all right? So bigger molecules can actually go back and forth, just free flowing across that channel through these transport proteins. They're not getting any use of energy, so it's still a form of passive transport. It's just diffusing through larger tunnels through the cell membrane. All right, so now we have active transport. Active transport, we have endocytosis. And in endocytosis, the cell actually takes material inside the cell. Endo for enter, things entering the cell. Now the cell has to be active. It has to use energy to move this stuff. So what it actually does is it opens up, surrounds whatever it wants to take in, and starts to bring it inside, okay? One type of endocytosis is this phagocytosis. Phagocytosis helps our body fight disease, and that happens a lot with white blood cells. If they find like a foreign invader, like a bacteria or a virus, they actually will surround that virus, that bacteria, bring it inside of itself and actually destroy it that way. Then we have exocytosis, and again, exocytosis is a type of active transport. The cell actually has to use energy to do this, and an exo, it sounds like exit. So exocytosis is the process of expelling or removing material. Material is exiting the cell. And again, the cell actually has to push it out. So it's pushing it out of itself. Okay, so that's exocytosis. All right, well, that brings me to the end of our presentation. We've talked about parts of the cell membrane, so the structure. We talked about the function, making a boundary and allowing some things to pass and keep other things through, being semi-permeable. So when we talk about the structure, we call it phospholipid bilayer, okay? And then it is semi-permeable, okay? It allows some things to pass, keeps other things out. And then we have passive transport and active transport. Passive transport, no use of energy. Active transport with energy. And then you guys can go through, we've got some examples of passive, which were diffusion, osmosis, and facilitated diffusion. And then our types of active transport, which were endocytosis, phagocytosis, and exocytosis. So please let me know if you have any questions, and you guys know how to get a hold of me. So I will see you in the next video.